Thank you.
All yours.
Thank you.
I had to talk to Alan yesterday, Murray.
Alan Morling?
Yeah.
Yeah.
And they have sorted out the water issue on the slides.
Oh, good.
But the slide thing is coming along slowly.
Have they got any?
I don't know.
Because coming along slowly means they haven't started.
No, they think about it all the time.
Yeah.
That's the impression. So I'm not quite too sure. You might need to touch base with him. I don't
So
know.
he's out at Royal Perth, isn't he?
Yeah.
So who's the best contact out at Fiona Stanley?
Fortunate.
Oh, Drew. Yeah.
Drew. Did you find out who it is at QE2?
No, I haven't sent. No.
I'll find out for you. I'll ask Yusra. She'll know.
Hello.
Hey, Emma. How you doing?
Good, thanks. How are
Yeah,
you?
good, thanks.
I was just noticing a strong separation here.
Murray, you're supposed to be on this side.
Oh.
I'm one for everyone.
You're one for everyone.
He's both exercise and haematology now. Hey, right. Hello.
Hey, how you doing?
Good, how are you?
Good. I've drummed up all the sports people.
Thank you.
Yeah. I'll introduce him as I come in. Mountain biker, shade road biker.
It's a really hard call.
Right.
Shade road biker.
Hello. Good, thanks. How are you?
Is that all right?
Yeah, that's fine.
So what's your role now? Where are you at now?
So I'm at the University of Tasmania still, so I'm in the finishing stages of my PhD.
What brings you here?
Well, Murray's my supervisor, so I've come over to do some intensive writing and some statistical analysis.
Right, so are you still writing up?
Yes, unfortunately. But originally from Murdoch, so Murray's organised office space for me and set me up really well, so I can get some work done. Yeah.
Just about ready to rock. Apologies, Alex over in the US at the moment. So
That's okay.
they're at the task of trying to organise.
Rein them in.
Decent crowd today.
Yeah,
So,
not
interest.
intimidating at all.
How do you pronounce your surname? Is it
Zadow.
Zadow.
Yeah, like dough.
Zadow. or Zadow.
No, it's Zadow all the time.
Okay, look, I think we'll get started in the interest of time. Look, it's great pleasure to have Emma here today. Emma is writing up her PhD, which is she's doing at the University of Tasmania, and is over here at the moment occupying a seat trying to quietly write up her PhD. And I'm sure many of you here have written up PhDs and know how important that can be. Emma completed both her undergraduate bachelor's degree in sports science and her honours degree entitled Physiological Responses to All-Out and Even-Paced Cycling Intervals, the shade here. She's a mad keen cyclist. at Murdoch University prior to moving to Tasmania. So she knows Murdoch pretty well. She's back here, which is great. And she's still got one head.
Yes. Hasn't grown yet.
And as I said, she's in her final stage of writing up her PhD, and her research interests obviously include pacing strategies and the influence of circadian rhythms within exercise performance and homeostasis and all things, Shay, related to cycling research. So I'm sure she's going to give us a great insight into how exercise should and shouldn't be done, I guess. And
We'll attempt
particularly
to.
from a cycling
We'll see how we go,
perspective.
yeah.
Okay, thanks, Emma.
Thank you for that introduction. So my PhD is entitled Athlete Health and Performance, the Role of Haemostatic Changes in Response to Exercise. So I'd like to begin by providing you with a brief overview of my PhD. So my PhD comprised of 3 studies with the main focus on exercise physiology and haematology. So study 1 was separated in 2 components. So the first component, sorry, can everybody hear me okay? Yep, perfect. So the first component comprised of assessing the validity of power output of a cycling ergometer. In this case, we looked at the Wahoo Kicker Power Trainer. Once we'd confirmed the validity of the power settings, we then looked at the reliability of a 4-kilometre cycling time trial completed with well-trained male cyclists. So study 2, we aim to investigate if coagulation and fibrinolysis are activated at different times of the day following short duration, high intensity exercise. And again, this is where the 4-kilometre time trial came in handy here. For study 3, we investigated a longer duration exercise bout, and we wanted to examine the effects of compression socks on haemostasis during a marathon. And for those who don't know the distance of a marathon, it's 42.2 kilometres. So it's a slightly longer duration exercise protocol. So moving on to our first study. So power output has been shown to be an important measure of performance. It enables us as coaches and sports scientists to accurately detect and monitor changes within competitive performance. And this is important as changes in performance as small as 1% often determine the difference between a finish on the podium or a finish within the peloton. And this is highlighted nicely by the 1989 and 2007 Tour de France wins by Gregory LeMond and Alberto Contador. Over 3,500 kilometres in a 3-week race, both won their respective Tour de France's by less than 1%. So being able to detect these changes within performance is really important. So cycling ergometers, as pictured on the bottom of the slide here, are important pieces of laboratory equipment used by us coaches and sports scientists. They enable us to assess overall fitness, whilst monitoring the effectiveness of training programs and the responses of ergogenic aid use, so the implementation of protein, caffeine and so on. Now, with cyclists reported to ride in excess of 35,000 kilometres per year, the ability for us to completely and truly replicate the dynamic demands associated with cycling is therefore highly desirable. And we tend to see increased replication when cyclist-owned bicycles are used. Therefore, regardless of the cycling ergometer used, any ergometer used in the assessment of performance needs to provide reliable measures of performance with ergometer errors of bias of less than 2% required. So what is the Wahoo Kicker Power Trainer? Well, besides being really fun to say, it's a mobile power trainer. And so it enables cyclists to attach their own bicycles, and this is through the removal of their rear wheel. And it's currently the preferred indoor trainer for professional cycling Team Sky. However, at the time of this study, the validity of the power settings of the kicker were yet to be determined. Therefore, the aim of this study was to examine the validity of power output settings of the Wahoo Kicker Power Trainer over a range of power outputs and cadences typically used within cycling. And for those who aren't familiar with cycling, cadences are known as the revolutions per minute, or how many times the pedal goes around in 1 minute. Okay. So the validity of power generated by the kicker was assessed by comparison with the power output of the dynamic calibration rig, which is this equipment on the right, my left, which is the gold standard in assessing the measures of power output for cycling ergometers. The dynamic calibration rig was used in order to control cadences across 80 to 120 revolutions per minute, whilst the power outputs were achieved by manually varying resistance for the kicker from 100 to 999 watts in an application for the Wahoo kicker. And so what I failed to mention on the previous slide is that the kicker can actually be controlled by an app for your iPhone or your iPad. And so this is how we control power output. So moving on to some of the findings, this figure presents all the individual responses over 100 to 999 watts at cadences of 80 to 120 RPM, with the overall average presented in the final column. Based upon previous research conducted by Abbas and Associates, ergometer errors of less than 1.5% were deemed as highly accurate, 1.5% to 2.5% as moderately accurate, and anything above 2.5% were deemed as highly inaccurate. And we've colour-coded this as green, yellow and red respectively for ease of interpretation. So if we look at the lower ranges of power output from 100 to 200 watts, we see a much larger ergometer error of 4.5%, unsurprising based upon the highly inaccurate readings at these lower ranges. However, if we look at powers ranging between 250 to 700 watts, we see a much lower ergometer error of negative 1.1%. And this is really exciting because it does fall within that recommended range for ergometer error of less than 2%. Again, unsurprising, at these higher ranges of power output, we see much larger errors of 13%, with higher or greater errors within 80 and 90 revolutions per minute. So in conclusion, when compared to the dynamic calibration rig, the Wahoo Kicker power trainer has acceptable accuracy. However, this is only over the power range of 250 watts to 700 watts. Caution should be applied for assessment of performance at the lower and higher ranges of power, and this is due to those larger ergometer errors. So we know that the Kicker provides valid measures of power output. So now we wanted to assess the reliability of a 4-kilometre time trial. So time trials require athletes to complete a set distance in as short a time as possible, and they're a highly reliable measure of performance, possessing coefficients of variations of less than 5%. Time trials enable us to monitor responses associated with training and fatigue, whilst providing a true replication of the bioenergetics required in competitive cycling performance. And you can't come from Tasmania and not include a picture of Richie Port doing a time trial. Okay, so here's our go-to guy. So this table here is presenting coefficients of variations for time trials ranging from 5 to 50 kilometres using a variety of different ergometers. And what's really interesting to note is that when cyclists use their own bicycles and attach to cycling ergometers, we see much lower coefficients of variations. However, whilst widely used within the literature, the reliability of a 4-kilometre time trial has yet to be determined. Therefore, the aim of this study was to assess the reliability of a laboratory-based 4K cycling time trial when completed on the Wahoo Kicker power trainer. So for this study, we recruited 12 well-trained male cyclists, cycling a minimum of 10 hours per week with previous time trial experience. Our participants were required to visit the University of Tasmania's Exercise Physiology Lab on 3 separate occasions, with each visit separated by a minimum of 2 and a maximum of 7 days. They were required to complete a 4-kilometre time trial and instructed to complete it in as short a time as possible, with feedback provided on distance covered only, so we blinded them to all other results. During these time trials, we collected measures of power, speed, cadence, heart rate and total time, with ratings of perceived exertion collected immediately post-time trial. So some of the results we found, so table 1 is presenting the mean and standard deviation for the 5 measures collected throughout and post these time trials. You can see we've got our results for time trial 1, 2 and 3, with our overall average presented in the final column. And what we observed is no significant differences whatsoever in any of these measures. Let's try that one again. So what I'd like to draw your attention to on this slide, however, is this overall average power output of 344 watts. So remembering that our ideal power range for the Kicker fell between 250 to 700 watts, we're pretty certain that power output is reliable for a 4-kilometre time trial when performed on the Kicker. So table 2, very, very messy, so I'm just going to talk you through it really quickly. So we're presenting the mean and 95% confidence intervals for within participant intra-class correlation coefficients and coefficients of variation. And what I'd like to draw your attention to is the overall means. So we're looking at power once again. So we see a relatively high ICC for mean power output with a relatively low coefficient of variation of 3.4%. Really good power outputs. However, if you draw your attention to cadence, speed and total time now, we see relatively low ICCs and relatively high coefficients of variations, suggesting that these aren't reliable measures of performance when assessing a 4-kilometre time trial. So therefore, in conclusion, the average power measured during a lab-based 4K cycling time trial is highly reliable in well-trained male cyclists. Okay? So this ensures that the 4-kilometre cycling time trial is reliable for monitoring changes within performance. Caution should be applied when assessing cadence, speed and total time, and this is due to those larger typical errors which we reported. So So this therefore leads us on to study 2. So we want to investigate if time of day and short duration high intensity exercise influence coagulation and fibrinolysis. So this is where the science stuff, as Murray would say, comes into play. So haemostasis is the body's normal physiological response for the prevention of excessive bleeding and clotting, and is constantly active at low levels until activated. So exercise, whilst beneficial for overall health outcomes, has been shown to have considerable effects upon haemostasis, resulting in a transient increase in blood coagulation, platelet aggregation, and fibrinolytic activity. However, this activation of these systems appear to be related to the intensity and the duration of the exercise itself. However, there are conflicting responses within the literature. So the activation of coagulation is measured by various markers of in vivo thrombin generation, with thrombin-antithrombin complex, or TAT, widely used within exercise physiology studies. Increases in TAT have been reported following short duration maximal exercise and longer duration strenuous exercise. However, within these studies, it appears that the intensity may be the most important factor within these studies. Now, the time of day in which these studies are rarely reported, and this may actually influence the overall coagulation response and the time of day in which the exercise is performed, which is really interesting. So, haemostatic markers have been shown or subjected to possess their own circadian rhythms, and we see an increase in hypercoagulability, hypofibrinolysis, and an increase in blood viscosity between 6 a.m. and 12 noon. This has therefore led to a transient increase in the frequency of thromboembolic events within the morning within at-risk populations. And by at-risk populations, these are people with predisposing genetic factors or metabolic or chronic conditions. This has therefore led to the recommendation that exercise should be completed within the afternoon only, with all morning exercise avoided completely. However, this does remain directed at these at-risk populations, with no current guidelines currently targeting elite and well-trained athletes. Therefore, the aim of this study was twofold. We aim to examine acute changes of coagulation following short duration high intensity exercise over 5 different time points. We also aim to examine if a time of day response is present within markers of haemostasis. And for this study, we chose to look at TAT, which is the in vivo thrombin marker of in vivo thrombin generation, tissue factor, which is the primary activator of cell-mediated coagulation, tissue factor pathway inhibitor, which is the major physiological regulator of the tissue factor initiated coagulation, along with D-dimer, which is a surrogate marker of fibrinolysis. So for this study, we recruited 16 well-trained male cyclists. However, we only present the findings for 15 cyclists, with one of my cyclists subsequently being diagnosed with a deep vein thrombosis 2 months upon exercise completion. So I'm lucky I didn't kill him. So always a bonus. So for this study, our participants were required to have had previous time trial experience, again cycling a minimum of 10 hours per week, and we did require them to be non-smokers with no known metabolic conditions, coagulation disorders, or previous thromboembolic episodes. So also throughout the duration of this study, they were required to not be on blood thinners or any non-steroidal anti-inflammatory drugs. So each participant was required to visit the University of Tasmania Exercise Physiology Lab on 7 separate occasions, again with each visit separated by a minimum of 2 and a maximum of 7 days. Participants' initial visit consisted of a maximal incremental exercise test, which I know Quentin is very familiar with, and involved beginning at an initial wattage of 70 and increasing by 30 watts every minute until they reach volitional fatigue. Okay, so this is what we typically put on our guys when we're completing a maximal exercise test. So you get a lovely headgear. We have a nice one-way valve here. We block off your nose. We collect your, fresh air comes in so you can breathe. And we collect your expired gas in order to determine your maximal oxygen uptake. And it is important to note that all these sessions were completed on cyclist-owned bicycle, again completed on the Waihu Kicker Power Trainer. So participants' second visit comprised of a familiarisation session, in which was used in order to familiarise our participants with the exercise protocol used within visits 3 to 7. Visits 3 to 7 were completed in a randomised and counterbalanced order and begun with an initial period of 10 minutes of supine rest. Immediately post this supine rest, we collected their very first pre-exercise venous blood sample. No rest for the wicked, they were straight onto the bike to complete a 10-minute warm-up, And this was standardised across all 5 experimental trials, with a peak power output as determined from the initial maximal exercise test. So immediately upon completing their warm-up, they were straight into the 4-kilometre time trial. And this was completed at either 8:30, 11:30, 2:30, 5:30 or 8:30 p.m. in standard laboratory conditions. Again, throughout this 4K time trial, we were lovely enough to provide them with feedback on distance covered only, where we collected perceived effort immediately upon time trial completion. So this was collected on a visual analogue scale, with 0 being no effort provided, 10 being gave my all. Okay? And immediately post-exercise, we collected that second venous blood sample. So moving on to some of the results. So figure 1 here, we're presenting the mean and standard deviation for power collected during a 4-kilometre time trial across our 5 different time points. What we observed was no significant difference within power output throughout the time of day. So figure 2, we're presenting the mean and standard deviation for our total time trial completion, our percentage of maximal oxygen uptake, heart rate, along with the effort provided. And what we observed was no significant differences within our total time for completion, along with heart rate. But interestingly enough, we did observe significant differences in our percentage of maximal oxygen uptake, regardless of no significant differences in total time for completion or power output. So we observed a main effect for time of day between 5:30 p.m. and 8:30 p.m. And you can see that the percentage of VO2 max was much lower here at the 8:30 p.m. time trial. We also observed a main effect for time of day within effort provided, with our participants rating the effort they provided lower at 2:30 and 5:30 p.m. in comparison to 8:30 p.m., which is really interesting. So moving on to the good stuff. So figure 3 is presenting the median and range for pre and post exercise measures of TAT, tissue factor pathway inhibitor, and D-dimer across all the 5 different time points. And what we noticed was that there was a significant difference from pre to post in all of our markers, regardless of time of day. We also observed a significant main effect for time of day for tissue factor, with much higher values observed at 8:30 a.m. in comparison to 5:30 p.m. in the pre-exercise markers. Whilst also not statistically significant, we did observe much higher measures of TAT for pre and post exercise at both 8:30 and 11:30 a.m. So this is suggesting that in vivo thrombin generation has started to occur. So in conclusion, a short duration high intensity exercise bout does result in acute activation of the coagulation and fibrinolytic system, regardless of the time of the day that the exercise task is performed. However, caution should be applied when completing short duration high intensity exercise at 8:30 and even 11:30. So especially in those with older athletes and those with acquired or genetic predispositions to hypercoagulability. So this therefore leads us on to study 3, in which we investigated the effect of compression socks on haemostasis during a marathon. So compression socks are widely used within the medical industry, and these are typically used to decrease the incidence of deep vein thrombosis following prolonged periods of immobility and surgery. So compression socks act as an external mechanical prophylaxis. They work by decreasing the cross-sectional area of the lower limb, therefore counteracting the components of virtuous triad. So we see a decrease in venous stasis, a decrease in coagulability, while seeing an increase in venous and arterial blood flow and velocity, ultimately resulting in a decreased potential for a deep vein thrombosis. Now, it is important to note that athletes are at an increased risk for deep vein thrombosis, and this is due to the associated demands with competition. So this exposes the athletes to repetitive microtrauma, endothelial damage, and dehydration. Now, compression socks are growing in popularity with athletes due to their reported enhancements of both exercise performance and recovery. And you've probably seen your footy players running around in a pair of skins. So they are growing in popularity. However, the impact of compression socks on haemostatic changes in combination with prolonged endurance exercise are rarely investigated. In fact, to our knowledge, we're aware of only one previous study to have investigated this, and this was a study conducted by Zaleski. They saw a decrease in thrombin-antithrombin complex in participants wearing socks during a marathon compared to a control group in which no socks were worn. However, the number sizes for the Zaleski study were quite low, with 10 participants within each group. Therefore, the aim of this study was to examine the effect of compression socks on haemostasis again looking at the same 4 markers that we did in the previous study when combined with a marathon run. So there was quite some logistics involved with this study, and it was quite exciting and quite scary at the same time. So we required the Gold Coast Marathon organisers to email recruitment flyers to all the runners who were registered to complete the 2016 Gold Coast Marathon. We weren't aiming for half marathon, we were aiming for full marathon, so 42.2 kilometre runners. From this, we had over 100 runners respond, and we asked them to complete a pre-exercise screening tour, and I had the lovely job of screening them all. So what I was looking for was the arrival time that they arrived on the Gold Coast, so I didn't want them coming in too early and I didn't want them coming in the day before, because we're looking at other aspects as well, which I'll discuss later. I also wanted to exclude runners who had had a previous history of deep vein thrombosis or pulmonary embolism, whilst eliminating smokers, people with metabolic conditions and oral contraceptive use. Also, my participants had to be willing to provide blood samples and wear compression socks. So some wanted the compression socks and didn't want the blood sample. So that then took us down to 80 participants who met the required criteria. From here, our runners were then randomly assigned to either the control group, in which no compression socks were worn during the marathon, or the sock group, in which compression socks were worn during the marathon. And from these 80 participants, we had 70 runners show up to the very first session, and they provided a written informed consent at the initial venous blood sample. And I just put Griffith and Southern Cross here, Southern Cross University, as we had students come and complete this for us as part of their practicum hours. So it was very nice of them to help us out. So moving on to the study design for this study, 24 hours prior to the marathon, so the Saturday before the marathon, we collected their initial pre-exercise blood sample. So you can see from the diagram, we were in a very small and cramped environment. We had approximately 3 stations up and running. We had 3 nurses provided from the Queensland Government of Health. I had 1 supervisor with me, Dr. Sam Wu, and myself collecting venous blood samples. So we collected these samples from 10 a.m. to 2 p.m. in order to minimise the influence of diurnal variations. And this was set up at the Gold Coast Exhibition Centre. So as the runners were coming to collect their marathon packs, we collected their blood samples. Those who were assigned to the sock group were then provided with their pair of compression socks provided by 2XU, and they were instructed to wear these socks on arrival to the marathon and throughout the marathon itself. So what we ended up with was an overall number of 36 within the control group and 34 participants within the sock marathon group. So a nice even distribution there. Now, immediately post-marathon, they were required to come and provide their final venous blood sample. So we were based at the first aid tent, which is approximately 100 metres from the finish line. So again, we were in very cramped conditions. We had 3 stations set up, and we lost 3 participants here because they didn't want to wait for us, and so it was a little bit late. Majority of our runners actually finished at the same time, which made it really difficult to fit everybody in. And the dehydration made those bloods very hard. So moving on to some of our preliminary findings. So this is all preliminary data. This is why I've come over to Perth to analyse some of the data. So what we found, we're presenting the median and the range for TAT, tissue factor, TFPI and D-dimer again, pre and post-marathon. So whilst not statistically significant, we do see much lower measures of TAT in those wearing socks during the marathon in comparison to those who did not wear socks throughout the marathon. What else was really interesting was that we noticed significant differences from pre to post in both the sock marathon and the control group for tissue factor and D-dimer, with an overall significant difference in sock group to the control group in the post measures of D-dimer. And our findings are so far in alignment with those of Zaleski, who previously investigated this. So in conclusion, haemostatic activation appears to be lower with the use of compression socks. The compression socks do not adversely influence markers of haemostasis, whilst compression socks may reduce exercise-associated haemostatic activation in individuals at risk of deep vein thrombosis. However, further research is definitely required. So some of the major outcomes that arose from the findings of my PhD are that the kicker does provide valid measures of power output. A 4-kilometre time trial is a reliable measure of performance, so it's great for all you cyclists out there. Coagulation is activated following a 4K time trial, regardless of the time of day it's performed. Care should be advised when prescribing short duration, high intensity exercise within the morning. And compression socks lower haemostatic activation and are safe for use by runners. So I'd just like to take the time to thank you guys for letting me come and present my findings, along with my supervisory team, Associate Professor James Fell, Associate Professor Murray Adams, Dr. Cecilia Kittick and Dr. Sam Wu, along with the Sports Performance Optimisation Research Team based at UTAS, along with my participants and collaborators. Thank you.
Questions for Emma?
Nope, that's fine.
Is it only for running, any other exercise too? Like
For the compression socks?
no, I mean the hemostatic marker elevation, or like the changes in those markers. Is it for swimming?
I guess it depends on the exercise mode. So research has shown that the mode of the exercise itself does influence the activation. So running has that nice impact on the floor, which initiates that coagulation cascade. Cycling tends to take that pressure off, but we do see an increase in blood pressure, which would promote that increase in coagulation activation. So that might be the same mode as swimming, with an increase in blood pressure potentially increasing from there. But that's rarely investigated. It's really hard to get those blood samples from swimming because they're wet. They don't want to. They don't want to dry off and stop. So, but yeah, that's a really good question. Thank you.
So in terms of the exercise, so are we suggesting you need to do more exercise in the evening than in the morning?
Yes, definitely. Yeah, we recommend that based on the previous research, and which is still directed at the at-risk population. They do say to avoid morning exercise, and we kind of show that as well. So we do see an increase in coagulation within the morning. So if you do want to exercise, I recommend the afternoon. But there's still further research required for that. So.
On the kicker,
Yep.
is the watts increased?
Yes.
Is that going up or down?
No, so the watts were increased. So we started off at 100 watts and we increased by 50 watts every 3 minutes.
So when you got up to like 800-900 watts,
Yep.
and it was showing out, was it less or more?
It depended on the cadence in which we assess. So it's really cadence dependent in the validity.
I'm just wondering because I can always get more watts out on the watt bike.
Oh, for sure.
And
And the limitation we had with the, sorry, I'm just sliding back.
I think with the kicker, when you're going up to a lot of watts, it's not as stable.
Yeah.
It moves about a lot.
Yeah, well, you can see in the higher ranges of power output, when cadence is increased, we do see greater accuracy.
Yeah.
So obviously, with a set resistance, the slower you cycle, the heavier it's going to feel. So that's kind of reflected in the results there. But
So
we,
that's 40% out or 68,
Yeah.
69% out to 80.
yeah, so huge errors. So I guess caution should be applied for those doing sprint interval training. So that's when you go all out, and power outputs typically exceed over 1,000 watts in these ranges. And so we know it's not going to be accurate for detecting changes within these higher ranges. But yeah, we could only go up to 999 watts. That was the maximal power output we could set. And we wanted to control as many variables as we can in that study. Hello.
Did you see any difference in results in your marathon runners when you looked at the female runners versus the male runners?
No, we didn't actually. I haven't actually looked at the differences in gender. I do have some other findings though looking at age as a factor. We typically ended up with approximately 22 females and the rest males, so we didn't have a nice distribution of gender, which wasn't the best. But I'll show you some of the other findings that we did have. We did do some other comparisons, only if you're interested, otherwise it's okay. Sorry, I'll go there. So some of these findings here, I don't know if you can see it properly, but we looked at the difference between domestic and international travel. We also looked at the mode of travel, so airplane versus other modes, so car, bus, plane. And we also looked at the differences in age. So we didn't observe any significant differences within domestic versus international, but we did see a difference within mode, which is really interesting. We'd expect to see an increase in TAT in aeroplane versus car. And we also observed significant differences within the age. So we did split them into 3 groups, and this was primarily for the marker of D dimer. So I don't know if that really answers your question, but we do have a couple of other variables that we are still investigating. But that's a really good one to look at. So.
Just on that, did you collect any sort of data on, say, the marathon runners and their sort of perceived effort?
Yes, so we collected pre and post marathon questionnaires. We also asked them how the socks felt and things like that, and we also took note of their nutritional requirements, what they've had to eat, etc. So that's still a lot of data that we have to go through at the moment.
Yeah, I was just wondering, like, some runners probably hit their target time, felt
Yeah.
reasonably good at the end, but others might have
Oh,
run out a bit
felt
faster and
terrible.
just crashed at the end.
Yeah.
Whether
Well,
that has any effect on.
oh, it could do. We did look at the finish times as well. So they had a mean average finish time of 4 hours 21 minutes. The fastest was 2 hours and 54 minutes, and the slowest was 6 hours and 10 minutes. And so it'd be interesting to see what their responses were to that. So see how we go.
The duration of travel, did you look at duration of travel?
We did, yes. So we looked at travel less than 4 hours and less than 6 hours, greater than 4 hours and greater than 6 hours, and we saw no significant differences. And that's in line with the domestic versus the international travel, where we didn't see any difference whatsoever. So I think the shortest time for travel was 1.5 hours, and the longest time was 18 hours. So, yeah, still lots more investigation to be done.
Obviously this has been done with, in inverted commas, elite athletes, but for the lesser mortals, I wonder whether, because, most of these kind of fun run events and things, they're done in the morning.
Yes.
Right? I mean, does that have any risk implications for those
Oh,
people
definitely.
who are not elite athletes?
Oh, definitely. You've got your weekend warriors who are coming out who aren't as well trained. They are at an increased risk for blood clots developing. So typically within your elite and your well-trained athletes, their blood vessels are well developed, so they're used to increases in laminar shear stress and blood pressure, whereas weekend warriors might not have trained. I love weekend warriors, don't get me wrong, but there may be an increased risk from there. So it would be interesting to see. We did look at their training volumes as well, and there weren't any significant differences. So if you did 2 hours of training per week versus 10 hours training per week, there was no significant differences. But again, that needs to be refined a bit more.
And just to add to that, I mean, is there I was wondering what the contribution of dehydration might be to the increase in coagulation versus impact on, time.
Yeah.
I'm wondering whether, because you're likely probably to get more dehydrated in the afternoon than you are exercising in the cool of the
Well,
morning.
definitely. So we did, unfortunately, we didn't control for that in the time of day study. We didn't control for plasma volume correction, so we had a piece of equipment, vital piece of equipment, malfunction, so we weren't able to obtain plasma volume correction for half of our participants, but we did have all plasma volume corrections for the Gold Coast Marathon. So, but yeah, the dehydration would play a key role, so, but they still exercise, so it was great.
I'm just curious, I guess, if you've got any insights as to, when you read your new pack of 2xU tights, it says hand wash and dry away from direct sunlight.
Yep.
The majority of people that I know don't do that, just chuck them in with the wash and off they go. If we're seeing any benefits from wearing the compression gear, how long realistically, I know you don't have any data, but if you've got any kind of insights as to how often people should be replacing their compression gear?
That's a really good question, Jack, but I honestly cannot answer that for you. We have a participant or a PhD candidate called Ned Brophy-Williams, who's pure PhD, is focusing on compression garments. He's based at the Australian Institute of Sport in Sydney. So he might be, if you look up some of his research, he might be better able to answer that. I'm sorry,
no,
but
that's all right. So I just
it's
feel like the benefits that you're probably getting from them are
reduced as it goes on.
People are wearing them 6 months a year after they've bought them.
Oh, definitely, and the way that they are sized are quite funny as well. So it's based upon your circumference of your calf and your foot size. And so if you have any great physical changes, of course you're going to need a different pair of socks, and they're not being replaced. So you might lose that compression aspect there.
For this study, though, they're all brand new.
Yeah, all brand new.
Yeah, I'm just curious about, like, if we're recommending you wear compression gear, and we're showing results, but then 2 weeks later after you've worn them 3 or 4 times, they're essentially the same as wearing a pair of socks. We just need to get that into account.
No more questions. I think we'll thank Emma for an interesting presentation.
Thank you.
Nice.
How do the socks stay up for 42 kilometres?
Oh, they
Do they ever fall down?
they stayed up pretty well.
That's
They
pretty tight.
they were really tight, and it was July, so it was a really hot day, and so they were not happy with us. They stunk really bad. So we got them in and out as quickly as we could. But no, they did overall want them to be as comfortable, and they did feel like it. We asked them if they felt like it improved their performance, and they did say yes. So it was like that placebo effect there, I guess.
Very nice. Well,
Thank you.
I have another question I'll ask. Totally unrelated to your talk. You're an accredited exercise scientist. Yes. Are you looking to get the accreditation, sports science accreditation as well?
Not really, because I'm not working with the sports teams.
And just because I'm on the working group for exercise science,
Yeah.
and I'm just curious as to why people.
Well, it's just, I feel like I'm going into academia, so if I was going to elite sport, I would definitely consider it.
Yeah.
But
No, just the fact that you've got the AES.
Yeah.
Because what we're finding is that so many people who are members of ESSA are AEDs.
Yeah.
And there's hardly
There's
anybody
no
who's
AES.
And so we're trying to grow that.
Yeah.
So every time I meet somebody, I just want to find out
Yeah,
if
no.
it's just an obligation, or
Oh,
if
no,
you
it's,
feel like you're actually getting a tangible benefit out of it, or...
Not particularly. Oh, sorry. Not particularly beneficial for me overall, because I was on the board for Tasmania ESSA, and we just got overlooked. There was 3 AES